I'm Dr. Marc Rothenberg here at Cincinnati Children's Hospital, and it's a great pleasure to share with you the advances that we've been making in the last year that help us understand and better treat eosinophilic diseases. This slide summarizes our research developments and what we've uncovered. This is a depiction of the intestinal epithelium, in this case focusing on the esophagus, and we now know that food antigens directly interact with the epithelium and can activate the epithelium to release cytokines, which we call pro-atopy cytokines, that are part of the innate immune system, such as these particular molecules, IL-33 and TSLP. These then activate the T cells and B cells to elicit memory responses that are being driven by the food antigens. These Th2 cells are releasing certain very strong cytokines or hormones that are influencing the rest of the reaction in terms of the allergic inflammation, and that includes IL-4, IL-5, and IL-13. And we've uncovered the signaling pathways that lead to these particular changes in the tissue, and this ultimately results in a loss of the barrier function, the integrity of the epithelium, the eosinophilic inflammation, changes in neurological sensation responses, particularly nociception, which includes sensation of pain and other symptoms, and that leads to different behavioral responses and triggers back into this inflammatory cascade. We also identified changes in proteases, which is an imbalance of antiproteases and proteases that are contributing to this response. And hence, elucidating all these pathways provides us with ways in which we can develop drugs to treat the patients. And if you just stay focused on this particular slide, I'll show you that we now have antibodies that allow us to block these cytokines, particularly IL-5, and ones that knock out and block eosinophils. And we just uncovered in the last year that this unfortunately does not improve the patients, but we definitively showed that the eosinophil is basically a biomarker of the disease rather than the cause, and this really changed the way we think about the therapy of these diseases. We also now know the importance of the IL-4 and IL-13 signaling because of drugs that are blocking this pathway. They're the first biologics that are approved for eosinophilic GI disease, dupilumab, for example, that's leading to great improvements in some of these diseases. We also know we have drugs that can block TSLP, and that's in advanced stages of development for these diseases. We can also block the IL-33 with a particular antibody. We can eliminate mast cells with a certain strategy that we're undertaking in patients now, and the results hopefully will be positive. We also know that we can use JAK, potentially JAK inhibitors, these are oral molecules to block this signaling pathway. And finally, we are working, and we have a study now where we're trying to restore the anti-proteases in patients, and we think that this is going to have an impact. So times are very exciting in terms of understanding this disease and really impacting the patients and helping improve lives around the world. I want to thank all the people that work together, my research team here at Cincinnati Children's, which include allergists and GI doctors, geneticists, pathology, coordinators, and research lab members. And also want to thank the funding organizers, certainly the NIH, the various institutes shown through the Rare Disease Clinical Research Consortium Network, the COFAR, which is the Consortium of Food Allergy Researchers, and then special thanks to the SIGA group, which is my colleagues that work across the U.S., and a real thank you to Ellen Kotroff and the Cured Foundation, because they are really providing us with important funds. And it's my honor to say that my lab is called the Rothenberg Cured Lab. And then the other individuals that are really as instrumental, David and Denise Bunning, Amy and Michael Ziccarella, the Buckeye Foundation, the Bornstein Family Foundation, the Louise Mayer Foundation, the Sebastian and Coulis families. So many other people that are helping us, and the patients that are helping us by telling us what their needs are and participating in our research. Thank you very much.